This presentation on PolyOrigin is sponsored by the Tools for Genomics Assisted Breeding and Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture within the USDA Specialty Crop Research Initiative, and was held on January 14, 2021.
Right. So, hi everyone. I'm Jeff Endelman from the University of Wisconsin, and I'm presenting today with Chaoji Zhang from Wageningen University. And the name of the software is called PolyOrigin. And we're now moving from looking at linkage in a single F1 population to considering multiple connected F1 populations. The background behind this project, in 2016, Chaoji led the development of this software called PolyOrigin, which we haven't presented here today, but it was the precursor for PolyOrigin, and it was not a full service linkage mapping population. It was really focused on the problem of haplotype reconstruction in a single F1 tetraploid population. But one of its main, two of its main attractive features were that the model for meiosis allowed for preferential pairing and quadrivalent formation in a very natural way. And it was available as a add-on for Mathematica, and I started to play around with it using our potato data and was impressed with the output. And that led me to contact Chaoji and ask him if he'd be willing to partner on a project to extend that methodology to jointly analyze multiple connected F1 populations. And basically what we're talking about here are partial diallel mating designs. So this is a figure from a paper from 2006, which just illustrates the different types of configurations that are examples of partial diallel. On the left-hand side, let me change my pointer over. So if we look at the sets of parents on the two sides of the square, if we have, say, all pairwise combinations, that's a half diallel, and you can have it either with or without selfing. This panel B here is showing what's called a circular mating design. If you took all of your parents and lined them up in a circle and you mated each individual with their neighbors, that would be this type of mating design. This is an extension of that. Part D is where the sets of the male and the female parents are two different individuals, sometimes called factorial mating or North Carolina 2 design. These are all examples of partial diallel mating designs. And from a practical perspective, these are the types of multi-parent populations that plant breeders work with, whether you're a polyploid breeder or a diploid breeder, compared to something like a magic population, which is really a specialized entity for genetic mapping research. It's not something that most of us probably are using in our regular breeding programs, whereas these should look familiar to anyone who's designed a crossing block for a breeding program. So in addition to Chaoji, we recruited Patricio Munoz, who's another expert on quantitative genetics in autotetraploids and leads the blueberry breeding program at the University of Florida, and his PhD student, Rodrigo Amadou. The four of us have been working for the last couple of years as part of a USDA AFRI project to create two different software packages that are now both available, and the manuscripts are under review, and you can read more details about them on bioRxiv. The one we're talking about right now is PolyOrigin, which is focused on analyzing the genetic marker data to reconstruct haplotypes in the progeny, and then that can feed into QTL mapping, which we're going to talk about tomorrow. So a bit more about PolyOrigin. The software is written in a programming language called Julia, which is relatively new, less than 10 years old. And it was designed by the computer scientists to combine the kind of ease of use features that you would find in a scripting language like R or Python, but have a high performance in terms of the numerical computation like you'd find in C or Fortran. And there are a number of ways that you can run PolyOrigin. You can run it from the Julia prompt. You can run a compiled version from either the terminal of Windows or of a Linux or Unix machine. And there's even a R wrapper that you can use to automate that. So you're going to see all those examples here in a minute. I'm kind of going through a bit of the highlights of how the software works and some results from simulation, and then Chowji is going to demonstrate the more hands-on part of using the software. The kinds of markers that this is designed for are biallelic, and we're envisioning either those come from a SNP array or from some type of GBS platform, and it could be a RAD-seq methodology. It could be target capture. It doesn't really matter. But the point is that the input data is either a set of dosage calls, like in the case of the array, that's what you would use, something like FitPoly or Supermasa to make those types of calls. And if you're working, however, with some type of sequence-based genotyping platform, really the best thing is to give the allele counts directly to PolyOrigin rather than using some other software like Supermasa or PolyRed. Rather than using some software to make genotype calls and then allele dosage calls and then feed them, you should use the read counts directly. And that's because the way the data are analyzed in PolyOrigin are on a chromosome-wide basis, and therefore it can properly weight the information that it gets from different markers based on the total read count. Whereas if you make the allele dosage calls independently, it doesn't know how much confidence to assign to those calls as it tries to accumulate the data across the whole chromosome. The software has models for how errors can appear in the data, both in terms of read counts and also the dosage calls themselves. So it accounts for that and can correct that information to give you corrected genotype data for both your parents and your offspring. And this software is designed to be used, I'd say, when you're at a little bit more advanced stage of working with your species than if you're trying to construct a linkage map for the first time. So if you're working with a single F1 population and you don't even have, say, a map for your markers yet, this is not the right software. This software assumes that you either have a reference genome that's a fairly good quality. It can have some errors, but it's fairly good quality, or you've already done some mapping with one of these other packages, and you may merge, let's say, the genetic maps from a couple F1 populations to get a consensus map. So you want to start with some initial ordering of the markers, and then PolyOrigin does have features to account for the fact that there could be errors in that ordering. So you can, at one level, ask it to remove markers that do not fit. And you can also ask it to attempt to do reordering of the markers. Although for some of the subsequent analyses with the QTL mapping, it's advantageous to keep the order of the markers in the reference genome and the genetic map the same. So at least in practice, I've tended to just use the remove marker option and not reorder them. So what do the input files look like for PolyOrigin? You have to give it a genotype input file and a pedigree input file. And here's an example for a half diallel with 3 parents. So there are 3 F1 populations looking at all the 3 pairwise combinations between these parents we're just calling A, B, and C. So the genotype file, you have the first 3 columns are your marker, chromosome, and position. And in this case, I'm using a reference genome to order my markers. Then the next columns are the allele dosage calls for the 3 parents. And then finally, we get into the subsequent columns, which are the offspring of the parents. And then the pedigree file, it just tells the software for every given offspring, what are the 2 parents of that particular clone. And the first rows are reserved for the parents of the population. So pretty straightforward in terms of the construction of the input files. They're CSV files that you give to the software. I'm not showing you an example of the input file for the allele counts, but instead of these dosages, you would have the two counts for the two alleles separated by that vertical pipe. All right, and what does the output that you get look like? One piece of output is the phased parental genotypes. So if we kind of toggle back and forth here, in the input file, we had the allele dosage for the three parents, and in the output now, for each one of those cells, it's going to tell us what is the phased genotype for that particular marker and sample. The alleles are named 1 and 2 in PolyOrigin, so that's why you see 1s and 2s separated by the vertical pipe, and that pipe is the separator for the different homologous chromosomes. So if you kind of look down and you stay on one side of that vertical pipe, you're looking at the genotype of one homologous chromosome. The dosages that we have here correspond to the number of copies of allele 2. So for example, this cell right here, which is parent A for marker M1, has a dosage of 2, and then we would expect to see 2 copies of allele 2, which here they are. Here's copy 1, here's copy 2. So this is very useful as you start to understand what are the haplotypes present in your parents. And then the second half is then understanding the presence and dosage of those haplotypes in your offspring. And that's what we mean by haplotype reconstruction of the progeny. This is a visual, this particular graph is generated using the diqtl R package, although there's a similar visualization tool in PolyOrigin, which you're going to see in a minute. But the point is the same, that we're looking at one particular individual. This is, let's say, offspring that we just called ID1. And this is the position in centimorgans along that one chromosome. And then each of the different rows here correspond to the parental haplotypes. And by haplotype here, I'm referring to a homologous chromosome in one of the parents. So if we're talking about tetraploids, each tetraploid parent has 4 haplotypes for this particular chromosome. And from the color pattern here, you can see the legend. The dark blue means that there is the presence of that particular haplotype in this offspring. And you can then look at this and you can easily see where the recombination breakpoints occurred. And we've seen examples of this already, so I don't want to belabor this too much, but just to look at this one example, this particular individual, you see it at this part of the chromosome, it contains haplotype, the first haplotype from parent A, and that's represented by a period in between the name of the parent, and then there's the period, and then there's a 1, 2, 3, 4 to designate the 4 haplotypes in that parent. So haplotype 8.1 transitions to haplotype 8.2 at this point of the chromosome. We can deduce that those 2 homologous chromosomes were synapsed and a recombination occurred there during the meiosis that led to this particular offspring. So that's one of the visualizations that we can have of the output from the software. Let's look at some other examples of this type of haplotype reconstruction. The software works not only for biparental populations, it works for uniparental populations. So in potato, you can easily self-tetraploids. Many of them are self-compatible. And this is an example of a real potato data set. We're looking at an S1 offspring. And so in this case, then we have both of the gametes came from parent A. And when you start to look at S1 offspring, you'll start to see that you have dosages that exceed 1. So now the light blue means there's a single copy of that haplotype, and the dark blue means that there's 2 copies of that haplotype. And again, you can kind of look at where the haplotype switches occurred, and you can infer something about which homologous chromosomes were paired and where recombination occurred in the meiosis that produced this individual. You can see dosages greater than 1 even in a biparental population when you have double reduction occurring. As I mentioned already, one of the strengths of the meiosis model that Chowji has built in TetraOrigin is present in PolyOrigin as well, is that it can explicitly model the presence of quadrivalence and then correctly infer the presence of double reduction. So here's an example of a real potato offspring, and we can see here that in this part of the chromosome, there are 2 copies of the haplotype B.2, And that means that both of the sister chromatids wound up in the same gamete in that part of the chromosome. All right, so that's kind of a high-level description of what you can expect to get out of the software. Let's say a bit more about how does it work. It uses a hidden Markov model, which you've already heard a bit about, and it does this across the entire chromosome, so that makes it quite powerful. And it proceeds in 2 steps. Step 1 is phasing of the parents, and then step 2 is the haplotype reconstruction. So in step 1, you have a bit of a chicken and egg problem. You're trying to decide what were the particular chromosome configurations, or what we would call valent configurations, that gave rise to the gametes that produced all of your offspring. And that depends on what the phased parental genotypes are. So the way the software starts by just making an assumption about the phased parental genotypes, and then given that assumption, it will find for each offspring what was the most likely valent configuration. It then goes to the second half of the iteration and says, okay, let's now assume that we have, we know what the valent configurations are for each offspring, and then what was the most likely phasing configuration for the parents, given that. So eventually it converges to a solution, and that concludes step 1. So then, now that we have both the valent configurations, the phased parental genotypes, and we have the observed marker data for the offspring, it's going to attempt to reconstruct each offspring in terms of the parental haplotypes. And it does that in a probabilistic way. And for each of the parental origin genotypes at a given marker, it's going to give a set of probabilities. This parental origin genotype is a bit of a mouthful. It basically is a description of the composition of the offspring in terms of the parental haplotypes. So for an F1 biparental population in the tetraploid, there are 100 possible parental origin genotypes, or different ways of composing an offspring in terms of the parental haplotypes. And the codes go from 1 to 100, and then on the right side of the table, you can see what that means in terms of the parental haplotype. So code 1 means that the individual has 2 doses of haplotype 1 from parent 1, and 2 doses of haplotype 5 from parent 2. The way that the numbering works is that haplotypes 1 through 4 are from the first parent, and haplotypes 5 through 8 are from the second parent. So this is a slightly different numbering configuration than appending a 0.1 or 0.2 and 3 and 4 to the parent name that I showed earlier. So this is the internal representation in PolyOrigin. And if you're talking about an S1 population, then there are 35 possible parental origin genotypes. And you can see they vary from having 4 doses of haplotype 1 and go on from there. So for each one of these parental origin genotypes, there's a certain probability for every individual and for every marker. Okay, so that's a bit about the way the software works and how it represents things internally. Now let's see some simulation results. And this is a snapshot of what you can find in the bioRxiv manuscript if you want to read it in more detail. This first figure shows the computational time and the phasing error for PolyOrigin compared to some other software packages. So to make a comparison both with TetraOrigin and with MapPoly, we're looking only at a single F1 population. If you look on the left panel here, the parental phasing error, that means using a simulation, we know what the correct phase is, and then we can see how well does the software reconstruct that. The error is very comparable between PolyOrigin and MapPoly, and however the TetraOrigin has a little bit higher phasing error because it doesn't have as flexible of an error model for the parents. However, if we come in and look on the right side and look at how long it takes to get that solution, PolyOrigin really shines. It's definitely the fastest of the 3 software packages, and that may be a combination of it being written in Julia and also some of the efficiency of the hidden Markov model algorithms that Chowji has used. All right, what about how do you want to design your your partial diallel doesn't make a difference in terms of the outcomes. So what you see here is kind of a complicated set of points, but the goal is to show you that across a wide range of designs, and what you see here is a linear mating design, a star mating design. Star is where you have one parent that's mated to a bunch of other parents, which would be similar to like a nested mating design like you see in these inbred diploid designs. Circular, the diallel, regardless of however you do it, you can explain the observed parental phasing error based on this metric here, which is the number of gametes from each parent that are present in the population. And so as you increase either the number of parents or the number of progeny, as long as you compute this number of gametes and you are over 20 gametes for that particular parent, you can be pretty confident you're going to have a very low phasing error. And it's actually a pretty reasonable number if you think about the kinds of population sizes we usually work with. 20 gametes from a parent is not a lot. So this shows that this kind of a analysis is quite feasible practically for many of us in our breeding programs. Then the next thing we looked at was, what about how you design your marker panel? And of particular interest to us was thinking about these newer types of genotyping platforms that many of us are considering now, where we can design the number of targets that we want to sequence. So it may be that it's based on some type of sequence capture with a hybridization-based design, or it could be these multiplexed amplicon technologies like the DART tag that some of us are exploring now. If you're trying to decide, well, how many targets or probes do I want? And then versus, well, how much sequencing do I need? There's some kind of trade-off, right? If you put more targets in the assay and you use a fixed amount of sequencing capacity, then the amount of sequence per target or the read depth per marker is going to go down. So what's the interplay between these different variables? And we looked at that using a 5x5 half diallel with 450 offspring and the balanced population sizes for each of those F1 populations. And that works out to 180 gametes per parent, which is a lot, way more than the number of 20 that we just talked about. So we have more than enough replicates to be successful with parental phasing, and it's therefore not surprising that the numbers you see on this contour map are quite low. The way to interpret this is that, again, you've got read depth per marker on the y-axis, number of markers per chromosome on the x-axis, and then the different colors show you different different constant values in this kind of a contour map, and the transitions here are shown with these numbers. So these errors are all very small, right? Less than 0.1% phasing error. So that's not the issue. The issue is, what about the error of reconstructing the haplotypes in the progeny? And this is where it becomes a bit more critical to think about the design of your assay. So a couple things to note here, right? So let's imagine first that you're looking in the vertical direction. You're going to keep if you say you had 250 markers per chromosome and you increase your read depth per marker, initially, as you go from say 5 reads up to about 20, you see some benefit in terms of the reconstruction error is going down, say from 0.15 to 0.08. But eventually, once you get above 20 reads per marker, you can see all these contour lines become fairly vertical, meaning that there's really no benefit to sequencing above 20 reads per marker to improve your haplotype reconstruction accuracy. It's much more important. You can see that the change in the error is much more sensitive to the number of markers. So if you look in the horizontal direction, if you increase from 250 and you go up to say 1,000, you see a noticeable decrease in your haplotype reconstruction error. So the bottom line is that if you're thinking about designing these assays, I would recommend you think about having at least 200 markers per chromosome. And you compromise a bit maybe on your read depth, and that's going to give you a better trade-off if you want to have an error that's less than 10%. All right, my last slide here is just circling back to this concept of double reduction, and to point out that just like Tetraorigin, that was one of the distinguishing features of it for modeling double reduction. This is a real strong point of PolyOrigin. We heard earlier that it doesn't make too much difference when you're trying to construct a linkage map, and that's true. But if you're interested in finding individuals that have higher doses of valuable haplotypes, and that's something we're going to demonstrate a bit tomorrow, this is really quite possible with PolyOrigin. This is a comparison on the top, PolyOrigin analyzing a potato data set versus MapPoly, which doesn't have a state explicitly for quadrivalence. And if you look at the dark patches, the dark segments here in the PolyOrigin, those are the regions where there's 2 copies of the haplotype. And those correspond to, if you look in the output from MapPoly, and this would be true for any software that doesn't have a model for quadrivalence, that you see that it tries to solve the problem by putting in a large number of haplotype switches, which is not realistic. And it's because it can't properly account for what's really happened biologically. So I'll stop there, and maybe what we can pause for a second and see if there's any questions about this part of it before I turn over to Chaoji and let him demonstrate the use of the software.
Hi, this is Yuman. Can I ask a question about genotyping design? You are thinking for the markers from genotyping side. For a breeder, I'm more interested in the phenotyping side. How many traits or what kind of traits I can include in this cross design, like qualitative traits or quantitative traits, how I can organize, or do you have any comments on that?
It should be applicable to any trait, really, in terms of any quantitative trait for sure. And actually what we're looking at here are just marker data. So we haven't even, there's no phenotype, no phenotypes involved in this part of the step. Tomorrow we're going to see how to put this together with the phenotype data, and I will cover both quantitative traits and qualitative traits as well. But these types of mating designs are applicable no matter what type of trait you're interested in.
Excellent, thank you.
Okay, I have a question. This, as I understand, you saying this software is only for the counting, I mean sequencing data and the array data, but not the SNP data. Am I right?
Not SNP data, did you say?
Yes. So can I use the SNP data here like BCF file or something, or we need something else?
Well, what did you use to generate the VCF file? What technology?
Most of the breeders, you having the BCF file or HapMap or PAD files, yeah? Not like the counting where you are saying you start from the countings and something. We don't have such files. So what is your suggestion if we have the BCF files with the SNP data?
Yeah, I mean, we need to be, the question that you seem to be asking is what kind of input files can be used, which is a bit different than asking what type of genotyping platform you're using, because a given genotyping platform can be represented, the results can be represented in different ways.
I'm saying the input files.
Yeah, so there is a certain input file format that you're about to see. And actually, I was just thinking about this yesterday, that we need to write a additional converter so that people can use VCF files as input for the software. I don't think we have that yet, but that's on our list of things to do.
I think that will be great for the breeders, because we have generally our mapping population we send for the outsourcing, and generally they give us the HapMap file or BCF file or PAD or BAD files. Yeah, thank you very much.
Sure, that's a good comment. Thank you.
Hi, Jeff. Tim Miller here. Just one quick question sort of about the limitation of biallelic markers. Is that a hard limitation of the method, or is that simply because the exploding complexity of the inheritance patterns if you go to multi-allelic markers?
Chaoji, you want to take that question?
Yeah, well, I think there are not many markers with multiple alleles, and I think the method is mainly designed for the biallelic markers.
Yeah.
I think it's quite safe to delete all the markers.
Yeah, okay. I'm just wondering if it's a hard limitation of the method, or if it's just a sensible limitation to avoid the complexity. But
It's not a methodological limitation, but it's definitely a software limitation. The software was written to only handle biallelic markers. Yeah.
Okay, cool. Thank you.
Jeff, this is John Cameron from Forge Genetics International. I was curious if the accuracy in the slide that you showed comparing the three tetraorigin, polyorigin, and map poly, if for polyorigin this is if this is specific to the diallel F1 case, or if this is a comparing single F1s.
This slide is comparing a single F1 population because that's the only type of population the tetraorigin and the map poly were designed to analyze.
Okay, I see. Great. Thank you.
Thanks for Jeff for the nice introduction to the project background and the concept and theory behind PolyOrigin. My name is George Zheng. In the following part, I will give a introduction to using the PolyOrigin in Julia and using PolyOrigin R in R, and give a demonstration. First of all, we need to install the Julia and also the Euro as a plugin for the atom. This is optional as an IDE. And for using the PolyOrigin in Julia, for using PolyOrigin R in R, then we also need to install Julia. But of course, we also need to install the R and RStudio. It's optional also, but I think it's nice to have it. And you know that in the Julia, there's three kind of repo modes. It's a Julia, help, shell, and the package mode. And you can type a square bracket, so you can from the Julia mode to the package mode, then backspace from back to the Julia mode. You can type this semicolon from the Julia mode to shell mode, and the question mark to the help mode. And we can install the Julia. You first type this square bracket to enter the packaging mode, then add the package from the GitHub web page. We can do similarly to install the PolyOrigin R in the R, then also install the PolyOrigin R package from the GitHub. Here this gives the link to the PolyOrigin web page and also the document. We can also update the PolyOrigin if there's a new version or new update from the web page. You can just type this square bracket up PolyOrigin. You can also get the help for the PolyOrigin function using this question mark. In fact, we try to build this package so that there's only one function. This is the only major function. So we integrate all the three steps. There are also other functions is mainly for the evaluate the result and visualize the result. So in principle, well, you don't need, but you can evaluate, check your result. Here's a workflow for using the PolyOrigin. First, we create the working directory. For example, we can name it as an example. Then you can put all your input file in this folder. So the PolyOrigin requires two input files. The first is genome file, is for the dosage data or the read count data, but also including the marker map. And the second is for the pedigree file. And then we can run the Julia. So run the PolyOrigin. In the Julia, the first mode, we first set the working directory using this command called cd. Then we load the package using PolyOrigin. Then we can run this with the function PolyOrigin, then provide these two required input file names. Then we also can set the options. There are a bunch of options, but mainly the two set of options is useful. First, if your marker map is a physical map, we need to say it is a physical map. Then we give this global average recombination rate so that you can have quite a reasonable total genomic length. Then we can set this refine map as true and refine order as false. In that sense, we we only estimate the intermark distance, but not the local mark ordering. Generally, if you have a large data set, then we prefer to start with a sub data set. Then we can select this subset of the chromosome or the linkage group, and also the subset of the markers. Here, something like here, we take array 5 marks. You can make this number is a little bit larger as long as it's larger than your total number marker. It doesn't matter. In the following, I will first go through the web page of the menu for the PolyOrigin, then go through a vignette for the real potato example. Afterward, I will show the real demonstration. Here shows the menu for the PolyOrigin. Then in the home page, we're showing that there's a simple introduction how to install the package and the reference. Then the menu shows the workflow. Get started with installation and using the package. Just basically load the package or using this kind of things for the parallel computation. The most important part to prepare the input files. You have 2 input files. Jeffrey already introduced it. So the first genome file is the first 3 columns is for the marker mapper, and the other is for the dosage. Don't go to the detail. I want to mention that the format of the genome type can have 4 possible. First, the dosage from 0 to 4 and missing, and the read count similar, and also the probability. From the dosage probability, it can be likelihood for the signal intensity from the sleep array data given all the possible of the dosage, or it's the posterior probability from the dosage calling program. In that sense, we are using the data twice. Also, it can be the probability for this rate count given the possible all the possible dosage. So it's actually generalized of this second rate count. And it's also possible to provide the parental genome type as a phased parental genome type. In that sense, the PolyOrigin will skip the step of parental phasing, then just perform the ancestral inference. The pedigree file is quite standard. We just provide the parents for each offspring. Then the founders of the parents are set to 0. For this run, PolyOrigin, I will go through the vignette. I would like to mention that this here is all the functions documented, but mainly you only need to look at this PolyOrigin function. Here is more like the help for this all the argument. And yeah, so I can, here is example, there's 3 examples as a vignette. First is for the simulated sleep array data, then is for the simulated GBS data. I will go through this. The last one is for the real potato data. Yeah, so we can set up the PolyOrigin for the parallel computation. So we need this distributed for the package, then you set the number markers. Normally we set the number markers as same as the number of chromosomes if you have enough number of CPU cores. And here this command, it means we make the PolyOrigin package available to all the workers. If you're using non-parallel, normally you just simply using PolyOrigin. Generally, we need to set the working directory. If you have a Julia file, you're simply using this CD, then this is, wow. We're just using this command to set the directory to the directory of the Julia file. So you don't need, if you copy the folder to any place, it should work. Then we set the input files, the name of input file. While we can also check the input files, we don't provide the notation tools, but you can just simply just read the file as a data frame. You can look at here for the pedigree and here for the genome file. We can also plot cross design. For this real potato data is a 3x3 allele cross. You can see here the name for the 3 parents. is a 3F1 population, named as the pop of 1, 2, 3, and here's the population size around 150. Then we can run the PolyOrigin. We can run the PolyOrigin for first 4 chromosomes. You can, if you don't set it or set it as nothing, then it will be run for all the chromosomes. Then you can set this auto stem as the ID for the output files. The first 4 option is to set the marker is a physical map. As I mentioned, here is the option to set the subset data set. This one is for the parallel computation. You can show the program saved 7 output file. Here, first is a log file. It's all the message print on the console. Here you can see it in the log file. You have this the version of the Julia and the version of the PolyOrigin and all the option values. If you didn't provide, then they will show it. It's easier to make sure later you can check what option you used to get result. You have also all the iterations used. And a lot then for the different step, I will explain a little more. Then it's all the computational time used. And the main output is a genome proper file. And also here the PolyOrigin file is more like a complete file. Then this 3 file is actually quite similar to the input genome file, except for this one that the marker map is refined. If you set the option refine map equal to true. And this second is a file is more like an intermediate result. Only the parent is effaced. If we I will demonstrate later. And this file is it's a positive probability file. It's more like if you have input from the GBS data, this is more like a multi-locus approach for the genotype calling. And if you have a sleep area data, I think you can compare this kind of posterior dosage probability with your input dosage to probably can find some kind of dosage error. But normally we only need the result from the polyancestral or genotype probability from the QDM mapping. We can check the result here. For example, we can read that is ancestral file from using this read this command. Then is basically this file contain multiple data frames. Here is a dictionary for the name of the different data frames. I just show you for example, this data parent genome, this data frame shows the phase of parental genotype. Here only shows for the 2 parents. And this is showing the genotype probability. I can only show for 1 offspring. Here show, for example, I show here for this markers, the certain marker, and for offspring this, named as this. And the genotype probability is provided as a sparse vector. Then the left side is the index of the ancestral genotype or the parental origin genotype. The right side is the posterior probability. It's quite sure then at this marker for this individual, the genotype is 64. But you can see we provided the full kind of posterior probability distribution. And here, another table is ancestral genotype, or you should call it parental origin genotype. Then it's more like the definition of this genotype index. Then showing here for this population 1, then showing this kind of the index for the genotype in this table. And also this the genotype in terms of the parental homolog. As I said, this is because if you have a different subpopulation of one population, then first the parent they are different. So that here list of the parent. Then the ancestral file also provides the posterior probability for the variant configuration for each chromosome for each offspring. I think Peter also mentioned that, but we only provide this is kind of the posterior probability for the variant configuration, so that for this individual in this chromosome, then this is possible, but the most likely is this one. So it's more like we have the, if we look at this, and this means this is a configuration for the first parent. Then all this for produce the gamete for this offspring from the first parent, and this part is for the second parent. Then they join two. This combined of these two configuration. This symbol means quadrivalent. Now you can see this symbol 1 pair 2 and 3 pair 4 homolog is a configuration for the bivalent pairing or bivalent formation. We can calculate the relative frequency of the valent configurations using this command. Then we provide this output as a data frame for at each chromosome for each parent. Then the relative frequency for this three, in total 4 possible of the configurations. The last one is quadrivalent formation. Then we can plot this one. It's like this is a plot for the relative frequency is averaged over chromosome. So then you can see the relative frequency for each parent. Then this color is showing for the quadrivalent. You see that quite around 1/4. Similarly, we can average over parent then to look at the relative frequency at each chromosome. You still see some kind of quadrivalent, of course. We can visualize the conditional probability or haplotype probability as Jeff shows is a bit different color, but I think quite similar. Then is the y-axis is for the 8 homolog or for the 2 parents. Then you can see here the red is more like close to the double reduction. You can plot all the RBD plot for all the offspring using this kind of the animation. And then it's very important. I think it's very useful for you to check whether your result is meaningful. You can compare your estimated genetic map with the input physical map to see it, for the estimated map. Now you can quite see clear here there's a centromere. Well, in the next, I will first to demonstrate so I'll first demonstrate to using the PolyOrigin in this Julia console. You can enter the Julia something like if you click here, then you got this Julia console. Then first of all, I assume that you already prepared this work folder, including this 2 required file. Then if you type in this mode, we first need to set the working directory. I already put here to set the working directory. You can see here is then we can run the command. Not here we're using PolyOrigin. Then run this one to set the 2 input files. Then of course you can set some kind of the options. Oh, here is a plot for the cross design. We have 40 parent for F1 population, 1 Sapien population. The plot for Sapien is not nice, but therefore that total 40 offspring, 3 parents. The PolyOrigin first perform the parental phasing. Then here is the iteration for the parental phasing. It's performed the 3 independent phasing. Then for all the runs, we get the same parental phase. Therefore, you see the difference is 0. That's 1 marker deleted. Then here's perform the unsettled inference, also including the parental error correction. Here you can see the 3 dosage that the 3 parental error are corrected. Of course, if you have a many, maybe I can show it. You can also give it to type this to get this kind of help for this. This is the package name. Then here is the function name to set. Well, you can also look at the website or the if you have a many kind of the prepare some kind of Julia file, something like here. Then you can using this like the sources, or here is a example you include. Then you can run all the Julia file in this command. Then it runs similar, just repeat these similar things. Yeah, it take about 40 seconds for this 40 parent one. Well, essentially we can look at this this kind of the example Julia file in this item. Here basically is here. I think I can delete it. Let me see. It's already finished. I just want to delete all the output here. It's here in the item, we see this kind of the in this PolyOrigin tutorial, we provide five example or then this example one, the folder, we have this example Julia. Then we can run it. If you type the shift enter, if you type control enter, it will not move to the next line. Then you can see here first. Then here we set the input file, then run it. Oh, here you get the result. Then also it runs similar. Probably I can make it larger. Yeah. It's basically you need just one line, or with this and this is enough. But you can also set a little more. Then afterwards, because this is the data simulated data, you can we can calculate the accuracy because we load the true values. Actually, you don't need this because here is the output log file. It's the same as you see here. Then we can calculate the accuracy. Then here it means that we can calculate the number of parental errors. Actually, here we get a perfect parental facing. There's a low parental error. Well, I think therefore we can make sure, quite sure this parental error correction is positive. And we also can calculate this ancestral inference error. Here is around 4%. It means that this sign error is 1 minus the probability of the true parental origin genotype. We can also here we can uncomment it using the control slash. Then we can run it for the to plot this relative frequency of the configurations. For this file is on actually this data file is simulated without double reduction, but you can still see a little bit of quadrivalent. I think mainly because this is a small data set. We only have 40 offspring. Similarly, we can run this to plot the RBD plot. Yeah, and you can, I think you can try all the others. The example, they have the Julia file. Maybe I should go back to the here. Yeah, just showing the screen cut for the demonstration. There are other examples. You can run your.
5 minutes.
Yeah. You can run it from the yourself for these three examples. You can read this HTML file, or it's, and for using the PolyOrigin, it's quite similar. We have a document and run the install the package. It's quite similar. The workflow is also similar. Additionally, we need to set the Julia path. Then you write the same, quite the same. Here is showing the example. Then if you run the PolyOrigin like this, then the result will be like this. I will not show it. I just looks like here is already run the result. The output is quite similar. Oh, I think, yeah. That's finally which one should I use? Actually, I think the PolyOrigin R is a wrapper for the PolyOrigin only one function. The other functions for the validation is not available, but the other ones they are same. Yeah, I think I'm done. I finished my part.
Does anyone have any questions for Jeff or Xiaoji?
Can you hear me?
Yep.
Xiaoji, just go back to the screenshot of your R Studio when you ran the PolyOrigin R. Slide 14. Yeah. I just want people to appreciate, I know you showed a number of the additional types of analyses that you can do after the fact, but this is the whole workflow right here. It really is pretty simple. I don't want people to come away from this discouraged that this is a complicated program to run. It's once you specify your files, you have one line 16. That's all you have to do to run the software.
This presentation has been sponsored by the Tools for Genomics Assisted Breeding in Polyploids Research Project. For more information, you can email us at toolsforpolyploids@tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.